Today's speaker is Professor Jean-Michel Cardot. JM is the professor and head of the Department of Biopharmaceutics and Pharmaceutical Technology at the Auvergne University in France. Prior to coming to the Auvergne University, he worked in the pharmaceutical industry for 15 years. JM earned his degrees in pharmacy, a master's in biopharmaceutical, statistical sciences, and pharmacokinetics, and a doctorate in pharmaceutical sciences from the Auvergne University. His research interests include biopharmaceutical development of drugs, in vitro dissolution, and in vivo bioequivalence, and in vitro and in vivo correlation. JM, welcome to today's presentation. I'll now turn it over to him to begin the presentation.
Hi, Susan, thank you. Hello, everyone. I hope that you will enjoy this presentation. It's going to be around 20-25 minutes. And at the end, as Susan said, I will be very happy to answer any questions. And if you have any questions later on, as Susan will tell you later, you can also ask it after. So, the first thing that we are going to do today is what is IVIVC. Then we will discuss about Level C IVIVC, in which case you can make it and how to do it. And we will see how to perform it in Phoenix and why Phoenix could have advantages over other softwares to perform IVIVC's Level C. I'll remind you something which is quite important is that IVIVC is a link of in vitro data to in vivo performance. And among the in vivo performance, it can be, for example, in vivo plasma concentrations or any type of in vivo parameters like Cmax and AUCs. Why do we have IVIVC? We have IVIVC not only to make mathematics or statistics, but to waive requirement to show evidence of in vivo bioavailability or bioequivalence based on in vitro data. The aim of IVIVC is to reduce the in vivo bioequivalence studies. That means it can be a surrogate of in vivo bioequivalence. And in this case, if we have satisfactory in vitro data, we can use them as a surrogate of the in vivo. And that is quite important to understand it because we discussed about in vivo bioequivalence, and we will see that it's namely two main parameters. So, when we have a plasma concentration curve, and we keep constant the active ingredients in quality source batch, the difference that we can see in the plasma concentration curves are going to be linked with the formulations. Or vice versa, if you keep constant the formulation and you modify the API, in this case, what you will observe in vivo is going to be linked with the quality of the API. A huge problem that you can have is to modify at the same time the formulation and the API, and in this case the problem would be that it's very complicated to distinguish between the formulation effect and the API effect. And very often it's more easy to optimize the formulation than to optimize the API. For IVIVC we have three levels. For the level A, we have a link between any observed in vitro dissolution and any observed in vivo absorption curve. That means, and you can see it in the left panel of this slide, that you have a regression curve and dots around it. Each dot or each triangle in this case is going to represent one time point. One time point means that the first one is half an hour in vivo versus half an hour in vitro, the second 1 hour versus 1 hour, and so on, for all the observed time points that you have in vivo. That does mean that you can make a regression with one formulation somehow, because in this case you are going to use all the time points. Of course, it's better and recommended by the authorities to use at least 2 or 3 formulations. What do you have on the right panel of this slide is the Level C IVIVC. In Level C IVIVC, you are going to put in relation some PK parameters. In this case, you can see on the y-axis is going to be the Cmax in vivo versus some in vitro parameters, for example, the person dissolved at the time X, for example, for a Cmax that can be 1, 2, 3, or 4 hours, or even earlier. And you see that in this case, each dots that you have and each marker that you have on this curve is going to represent one formulation. and that is quite important to understand it. From a regulatory point of view, we say that the level A is more powerful than the level C because you use all the information that you have on the plasma concentration curves. At the opposite, the level C is said to be less informative. But if you have a level C which can correlate the Cmax to some in vitro dissolution parameters, and at the same time the AUC to some in vitro dissolution parameters, that means you are able to predict all the PK parameters which are dealing with bioavailability and with bioequivalence. In this case, it's quite important to understand it because you can use in some specific cases a surrogate of in vivo study also because you can predict the bioavailability of the curves. Sometimes, even if level A is said to be better and you must try to do it, sometimes you cannot make a level A IVIVC. You cannot make a level A IVIVC, for example, when you use some metabolites data. That means you have the enhanced drug which is administered to the patients and you are going to measure in the blood the metabolite because it's quite complicated to measure the enhanced drug or the metabolite is active and the enhanced is inactive. Even if, according to bioequivalence guidelines, you have always to measure the parent compound. Why, in this case, it's quite complicated to make a level A IVIVC? It's complicated because the metabolite can be formed pre-systematically. That means before you can dose them and you can analyze them in the blood. And in this case, it's going to be formed in the liver, for example. But also metabolites can be formed during the elimination process from the parent compound which is present in the blood via the liver. So what you can say as the absorption curve is not representative of the input because it's going to represent the input and also the formation of a metabolite through the normal liver metabolizations as a route of elimination. The second case when it's not possible is, for example, when you cannot make deconvolution and you are not in one compartment but in two compartments. That means that you cannot have any input function UIR in Phoenix due to the fact that you don't have any solution or IV curve. So in both cases, you can try to make a level C, and the level C is going to be a very good alternative. It's going to be a good alternative because you cannot perform any level A, because it's better to make a level C than nothing. And if you make a level C based on the Cmax, AUC, or other parameters which are of importance, you can predict not the full profile of your plasma curve, but the main parameters which can help you to predict bioequivalence. So it's very important because you will have all the BA/BE parameters. In some cases, you can even try to use it as an in vivo surrogate. Of course, some authorities can refuse it, or some authorities can tell you that it's not in the guidelines, but in specific cases, they could also accept it. You must use only the test formulation having the similar release mechanisms and technology. That means you are not allowed to use the reference formulation or the formulation of a competitor to establish a level C IVIVC and then try to use and to extrapolate the result to your own test formulations. So, if you want to perform level C IVIVC in Phoenix, you must calculate PK parameters like Cmax for the rate, AUC for the extent, and any other type of parameter. For example, if you have for antibiotics or for some antiparasitic drug, the time over the minimum imidari concentrations can be a very important factor. You must be able to link them to dissolution data. Then you must find the best relationship, and we assume very often for level C, like for level A IVIVC, that is a linear relationship, and you must calculate the predictability. So you have a lot of steps that you must perform, and you can make all of them in Phoenix. So you can also calculate the IVIVC on all derived data. That means if you have 24 subjects, 3 formulations, you can use the 24 Cmax and AUC for formulation 1, the same thing for formulation 2 and formulation 3. That means you will have a lot of data. The interest to use all the raw data is that you will have a better idea about the viability of the results. Of course, when you are going to predict a new Cmax and AUC based on new dissolution profiles, in this case you will have exactly the same variability as the variability that you have observed for the initial data. The second point, which is also of importance if you use all the raw data, is that you will have by definition a lower coefficient of correlation R. Why? Because you have more data. But the robustness of your correlation is going to be better. You can calculate VI, VIVC on the mean data. It's a straightforward, simple process because you minimize the number of points. Results in slope, intercept, and predictability are going to be almost the same between the two. I will use some data which are simulated data. The idea is to use a pilot study with 12 subjects with a failed results. The four formulations are for the same sponsor. You have a current formulation that is called R. It's rare R because it's the current formulation, so it's the reference formulation for the sponsor. And the three alternative formulation A, B, and C, all of them are at the beginning said to have the same technology, both test and reference. That means the same release mechanisms. You have two dissolution methods available. Why two? Because you make a first one, and as you failed your B study, that means you have to think that maybe your dissolution method was not the most adequate. The data are published and you have a reference in the bottom part of this slide. Of course, not exactly in the same type of calculations between what is published and what is presented here. So you have here the results. You can see that the reference is higher and the tests are lower. You can see that the test A and B are very close together and the test C is very slow. In vitro, you have the first in vitro dissolution method, which is on the left part of this slide, and you can see here that the A was fast, the C was slow, and the B was on the middle, almost overlapping the reference, that is the current formulation on the same sponsor. As we failed the B study, that means also that the in vitro dissolution method 1 was not correct. Why? Because it do not reflect exactly the same difference that you have observed in vivo. So after, because the B study failed, they had developed a new dissolution method where you have the right ranking between the formulation and what you observed. A and B are very close together, C is lower, and reference was faster. Only to show you what is the problem. If you use the dissolution method 1, which is on the left panel here, you can see that you have a R square which is not good. Why? Because you can see that all the formulations are not matching, and not matching in distance between formulations, but also in all the formulations on the same straight line. At the opposite, if you use the dissolution method 2, you can see that the R square is better and is better matching. So its dissolution 2 seems to be predict better than dissolution 1. But that is the first thing. Now we have to see how we can achieve both type of graph. So how to perform it? You can start with all data. It's more complex, and it's exactly what we are going to do now, only to show you. You are going to use everything to organize your raw data of plasma concentration and dissolution. For example, you are going to use stacker and to stack if necessary. Then on the plasma concentration, you are going to use non-compartmental analysis, NCA. You are going to use also the join to join the dissolution data with the PK data. The linear module also to see what is the best relationship. Data wizard to extract the best relationship, then pivot to put it in the right place, descriptive statistics, then the new time data wizard for predictability, and plot. So you are going to use a lot of functions. Step by step, what are we going to do? We imagine here that your raw data plasma concentrations are in the right format, and that it's quite easy to see. Now you have to calculate all the PK parameters of interest. And often first you are going to calculate Cmax and AUC because it's the two bioequivalence and bioavailability parameters. You can use any other type of parameters as surrogate of activity, and that I made it. For example, you can have the first time below the effective minimum concentrations when it's, for example, for some drugs which are related with a very good PK-PD relationship, or time over the IMC, which is the inhibitory minimum concentrations for antibiotics. But you have always to justify the type of parameters that you are going to use. If you work on the mean value, you have first to calculate the PK data, then to use the mean and descriptive statistics to go ahead. Now, when I have performed and I have calculated all my non-compartmental analysis, what I have to do, and that is more important, I have to add the dissolution data to all PK data. I am going, if I use all the raw data, between the NCA results and the dissolution, I'm going to use a join with the right key, which is going to be the treatment. So you are going to join worksheet 1, which is the NCA final parameters pivoted, and the worksheet 2, which is the in vitro data, and in this case of the dissolution 2. You are going to sort them by treatment, and then you are going to select as source columns, that is the in vitro data, time, and dissolution. And for the in vivo data PK parameters, it's going to be Cmax and AUC, for example. And you are going to have a result, which is the table, which is in the below part of this slide, where you have for all treatment the Cmax by subject and the AUC by subject. And for the same Cmax and AUC and subject 1, you are going to have all the times and all the dissolutions. You are going to have the same thing for the subject 2 treatment A, and the same thing for the treatment B, C, and the treatment which was the current formulation on the market. That means you will have the 48 treatments times subject line multiplied by all the dissolution points. That means you are going to have a huge table. Based on the huge table now, we must extract the best pair for the Cmax and or the AUC of match between the Cmax parameters and the dissolution. You are going to use the regression is going to be the linear model regression. And you are going to perform it by time. And you are going to use the linear regressions because if you have a nonlinear relationship, that could be a problem. So you are going to use the join raw data and dissolution. You are going to sort by time, and you are going to put as X value the dissolution and Y value, for example, in this case, the Cmax. And you are going to have the results, and the result is going to be a table with by time, the best relationship, and the error factors, slope, and intercept. And you are going, of course, to extract the best error value, which is going to be your best relationship. You are going to select the diagnostic panel in the results, and in the diagnostic panels, you can see that you have a lot of parameters and you can use correlation. In this case, this parameter is going to be a good approximate of what you have to use. You can use something else also. And you can see, for example, in this case, that for the time 0.5 hours, the coefficient of correlation is quite low. It's quite low, that means it's not the best one. You are going to pivot it. You are going to pivot it now. Why? To have the time, the criteria, and the coefficient of correlation. That will help you, in this case, to extract very simply the best coefficient of correlation and the related time. That means the maximum coefficient of correlations and the time to have this maximum coefficient of correlation. To do so, you are going to use something which is very strange, which is the non-compartmental analysis. Why? Because in non-compartmental analysis, you can calculate, of course, all the PK parameters, but you can calculate a pair of parameters which is very important, which is the maximum value of something and the time to have it. When it's plasma concentration data, it's called Tmax and Cmax. But if you replace the concentration by the coefficient of correlations, in this case, that will extract the maximum coefficient of correlation and the time to have it, exactly what you need. So you are going to select, in this case, in the NCA, the time is going to be time, and the concentration is going to be the correlation coefficient. You are going to go then in the parameter name panel, and you are going to change the Cmax by max R, which is the maximum coefficient of correlation. You can see it in the below part on the right part of this slide. And when you run it, you will have extracted the maximum R, which is the maximum coefficient of correlation, and the time to have it. So for the Cmax, you know that the best time to have a linear relationship is 2 hours, and that the coefficient of correlation is 0.8755. When you have now the time and the maximum coefficient of correlation, you have to extract all the other data which are of importance. That means you are going to extract, in this case, all the data with the Cmax and dissolution, and you will have also to extract the slope and the intercept. Again, you are going to use join, data wizard, and then plot. You are going to use the join. That means you are going to join the results of the NCA, that means the Cmax, which is the maximum error, and the time to have it, with, for example, the join data between the Cmax and the dissolution. So you will have something which is, in this case, going to be a table in which you will have the time, the maximum error, which is the maximum coefficient of correlation, the Cmax, the treatment, the dissolution value, the subject value. You can see that when I join it, you have time in which maximum error is equal to nothing. Is when you don't have it, that means not the time at which you have the best correlation. Then you can use the data wizard on both results. If you use the data wizard on both results, you are going to use a filter specification, and you are going to exclude the value of maximum error equal to blank or null. So you are going, and you can see it on the right part of this slide, bottom, exclude null in column max error equal, and not below or greater, but just equal. When you run it, you will have then a subset of data which correspond to the time, the max error, of course, the Cmax, the dissolution, and the subjects. So you have everything that you need now to make a plot and to calculate your predictability with the intercept and the slope that you will have also to extract. So now that I have my subset, I can make very simply a graph. In this graph, I will use the dissolution as the X, and I will use the Cmax as the Y. You can see in this case that I have a very nice graph on the right part of this slide, and you can see that I have a good relationship. This straight line which corresponds to the relationship, I can have it in my plot options. In my plot options, you can see that I have graph. Under graph, I have Cmax versus dissolution. Then I have error bars, A, B, C, which are all the treatment, but I have a first line, which is regression. And if I click on it, I have a panel in which I can generate the regression. I select linear, and I show the associated title. That means in this case, you can see on the top of this slide that I have the R square, not the R, but the R squared. That means R multiplied by R, which is here, and the intercept and the slope. So even on this graph, I have now my relationship. But this relationship cannot be used straightforward to predict, because that means I will have to extract the value on the intercept and the slope and re-enter them by hand in the software, which is not good because I will not trace everything that I have done. So what I have to do now, I have to use it for the predictability. What I am going to do, I am going to use my summary table. I'm going to use my descriptive statistics to extract my Cmax and my residuals versus time. Why? Because my residuals is nothing more than the difference between what I observed and what I have at the origin as predicted data. So that is in the linear model summary. If I pivot it with variable as column header and row header the time and treatments and the mean as value and everything, I will have in this case directly the predictability. Predictability is going to be the residuals, and that I can do it in data wizard, is going to be the residuals divided by the Cmax multiplied by 100. And so I will have directly in this case the predictability for the 3 formulations, and I will make everything directly. Next point which is important to calculate the mean predictability is to use the previous data and with the descriptive statistic to extract the mean. So I am going also now, I can make something which is going to extract from the data the final parameters, because that is a way also that I can use then to predict everything. So I go back in this case in linear regression. I am going to extract in the final parameter pivoted, I am going to extract the intercept and the slope. And I'm going then to join it with the maximum error as just I have made before. And in this case, that will allow me to select the line in which I have the same time, the intercept, the slope, and the maximum coefficient of correlation. Extracting that, I will have in this case my slope and intercept directly in Phoenix, and I can use it to, if I have any in vitro dissolution data, to predict the Cmax, for example, or the AUC of a new dissolution curve. So when I don't want to use the individual data, I can make the same thing directly on the mean value. The only difference will be that I'm going, in this case, to make all the same steps, but I don't use the individual data, but the result of the descriptive statistics of the NCA directly to perform my Level C IVIVC. In this case also the second difference, as I'm going to have the results, is that I will have to use also as a group parameters the variable, the column variable, to have the difference between the AUC and the Cmax. In this case, the keys to join them are going to be variable and time, and not only time. If I make it on all the raw data, or if I do it on the mean, you can see that I don't have exactly the same scale on the, in this case, on the y-axis, but I have exactly the same intercept and the same slope. Of course, the S-square is not the same one, because when I have only 4 points, I have a better coefficient of correlations and R squared, of course, but you can see that the intercept is 0.368 on the left part of this slide, and I have exactly the same thing on the right one. On the right one, I have a slope of 0.04992, and you can see also when I use all the data that I have the same slope, which is normal. That means using the mean data or using the individual data do not bring any difference. The only difference is when I use all the raw data, I have a better idea about the dispersion of my Cmax between subjects and not within subjects, between subjects. If I calculate using the residuals variability, you can see that the variability is exactly the same if I use the mean data or if I use the individual data. The predictability, for example, for the formulation A, you can see between the upper part and the lower part of this slide is 2.16668%, and I have exactly the same value when I use all the data. If I take the formulation R, it's 2.001988, and I have exactly the same thing. You can see it on the top part of this slide, last line. Why? Because I am going to make the predictability on the mean value, and making the mean value before or after making the regression is not going to provide you any differences. So what is the advantages to use Phoenix? In Phoenix, you can calculate automatically the NCA. You have a linear regression also, which is included inside. You can extract the data using Data Wizard and join, which is not so complicated. You can trace all the calculations. That means you have only to enter the time concentration and the time dissolution, and then with all the subroutines which are existing like join, Data Wizard, linear regression, plot, you can directly make your Level C IVIVC without manipulating the original raw data. That will secure all the process because you can trace everything, and when you have a result which seems to be a little bit strange, you can see where you have, for example, forget to select variable or to select the right time. You have a variability of calculations because we discussed about linear regression, but you can use other type of regressions. You can also input a lot of things. For example, you can modelize your dissolution curve and see if you don't have a best sampling point than those that you had at the beginning to make an IVIVC. However, you have to program and to use the right equation. That means you don't have this facility directly in the IVIVC wizard. That means you must know how to use all the steps of Phoenix. In the steps of Phoenix, which are important, you have everything related with data, everything related with non-compartmental analysis, and with linear modeling and linear regressions. To conclude, you can have always a nonlinear scaling. That means you can use it, but you have to know why you use it as a nonlinear time scaling and nonlinear scaling. You can use it only when you have a good possibility and when you know why you use a nonlinear scaling. You can, the second problem that you have is that you reduce the full profile to one or two single values. That means the full profile of plasma concentration is going to be reduced only to a Cmax and AUC. That is a drawback of all Level C IVIVC. But a Level C IVIVC is better than nothing, and it can be used as a first approach. If you have a Level C correlation on both AUC, Cmax, and maybe Tmax and so on, very often you have a Level A somewhere, and you have to investigate this possibility. You have to think that you must sometimes redevelop a dissolution test. And you have to be very careful in this case when you redevelop it to make something which could be used also as a QC. Why? Because if you use something which cannot be used as a QC and the authorities see that you have an IVIVC, they can force you to use the dissolution method that you have used during your IVIVC as a QC tool. So, do you have any questions? I am open to all of them.
Great, thank you so much, JM. Please do enter your questions for Professor Cardot into the Q&A box, and we will answer them. First question, JM, from our audience, someone would like to know, do you prefer using arithmetic mean or geometric mean when running Level C correlation on mean data?
You can use mean data or median data also. Arithmetic mean or geometric mean, the difference is not very important if you don't have any, into quote, outliers. Arithmetic mean is very sensitive to extreme value, extreme low or extreme high value. I would say that you can use both of them. If you want to be more stable, use geometric mean and median because it's going to work over all the time quite easily without any problem. If you want to use geometric mean, you have to take into account that if you use all the raw data and then you make your IVIVC Level C, in this case your IVIVC Level C is going to go more through the arithmetic mean. But that is not a major order because you have some other possibilities to program your linear regressions. But so if you want to use mean, I will go more for geometric means or median. Median is a very good option also. And geometric means is more close to what you can calculate also in B study because you make for B study a lot transformation.
Someone would like to know, when is a Level C IVIVC useful compared to a Level A IVIVC?
So a Level C IVIVC can be very useful, first of all, when you don't have enough data or you don't know if it's possible and you don't want to investigate time. You can try very easily at the beginning with the mean value of Cmax, AUC, and Tmax, for example, even if you want to see if you have a match with your dissolution data. If yes, and it's matching for the three parameters, then you can think that you can invest time to make a Level A. The second case is when, for example, you have a prodrug and this prodrug has very limited plasma concentration compared to the active metabolite. And if you have, in this case, very limited plasma concentrations, the problem is that the metabolite is going to reflect the initial pre-systemic metabolization, as we discussed a little bit before, but also the current elimination for the plasma. And it will be misleading sometimes to make directly a level A IVIVC. And in some cases, you don't have enough data. For example, you don't have any IV data to make deconvolution, and in this case, you are forced somehow to make a level C IVIVC. That would be, I think, the three main cases in which a level C IVIVC is quite indicated. But as I told at the beginning, a level C IVIVC can be a very good starting point when you don't want to invest time to see if you have any chance to have a correlation.
Can I perform a level C IVIVC with one or two formulations?
Not really. You can do it for level A, because on level A you use all the plasma concentration data. But as in level C, you restrict your plasma concentration curve on the for one point, which can be the Cmax, for example, or the AUC. If you have only two profiles or two formulation A and B, and you restrict it to Cmax and AUC and only to the mean value, of course you will have a straight line between the two mean value. That is one of the drawbacks of the level C IVIVC, and for this reason it's said to be less powerful, because you are not going to use all the data, but very often you are going to restrict your data to only one single point, which is going to be the mean value.
Looks to be the last question from our audience. Could level C IVIVC be used as a surrogate of in vivo bioequivalence?
Officially in the guideline is said no, but I know that some authorities accept it mainly when, for example, you can correlate all the bioavailability parameters, that means all the Cmax and AUC's values with dissolution. That means you can predict the risk to be non-bioequivalent. In this case, it can be used as an in vivo surrogate, and it was accepted by some authorities. One advantage of the level C in this point of view is that you don't have to perform any, for example, deconvolution. That means as you use directly the Cmax and AUC, and you have not to perform any deconvolution, it can be a very simple, straightforward way to go. But if you have all the data to perform a level A, the authorities are not going to be very happy.
Looks like we have a few more questions. Whenever we do dissolution testing, the time points don't match. The time points in the dissolution test don't match the in vivo time points. Can we do an IVIVC for such cases?
Of level A, no. Of level C, yes. Because on level C, as you restrict the plasma concentrations to Cmax and AUC, you don't have any notion of time. That means you don't need to be at the Tmax to make the correlation between the Cmax and the dissolution. You only try to see which are the dissolution data which are going to fit with the in vivo data. At the opposite, when you want to make a level A, you need to have some data between dissolution and in vivo which are going to fit, or you need to make a modeling of your dissolution data to extract the time points which are going to fit after a time scaling, but only after a time scaling in this case very often.
Looks like our final question is, when do we need to perform deconvolution in IVIVC?
In level A IVIVC, you need to perform either a deconvolution, a classical numerical deconvolution, or a Wagner-Nelson or a Lurie-Goldman deconvolution, if we can call that deconvolution, to extract the absorption curve. That is for level A. That is not for level C, because in level C you are not going to work on the absorption curve, but you are going to work mainly on the PK parameters. So deconvolution is for level A, not for level C.
Okay, thank you so much, JM.